Welcome to MOOC course on Introduction to Proteogenomics. In the last lecture by Dr. Bing Zhang, you were introduced to the concept of network analysis with emphasis on protein-protein interactions. In today's lecture, you will be explained the need for network visualization and the various resources available to make sense of the complex data. I am sure you appreciate that not only analysis, but also the data presentation and visualization is very crucial. And in this light, Dr. Bing Zhang's today's lecture is going to provide you good insight and opportunity to look at various tools available for data visualization. So, let us welcome Professor Bing Zhang for today's lecture.
Next, I'm going to talk about for our data analysis and what can we use this network to help us. And one way is we use this to visualize for visualization, and the other way is to do some data analysis. So I'll talk about the network visualization first. So network visualization is usually through the node link diagram, as we said. And so each gene or protein is a node and they are connected by an edge. This is called the link, so it's called node link diagram. And the beauty of this is not only you can see this, and then you can overlay data to the network in order to understand your data, right? For example, I'm interested in a certain type of proteins in the network. Let's say I'm interested in transcription factors. And then I can change the shape of the nodes to a triangle shape, and then immediately I say, okay, those are transcription factors. And then I can overlay my differential expression result to the network, and then we can say, okay, these genes are upregulated and these are downregulated. So, and there are tools that have been developed to facilitate the network-based both the network visualization and the network-based data visualization. And this review article in Nature Methods summarizes a lot of tools that you can use to visualize biological networks. And this is, the most popular one is Cytoscape. It's very easy to use and very powerful. This is one network I did in an earlier publication. And if you you can also put some artistic change to the network. I mean, actually, this was also done in the Cytoscape. And after I did this, and this became a cover of that issue of the journal. So within the Cytoscape, you can actually do a lot of cool things to visualize your network and your data. But there is also a challenge. I mean, although the Cytoscape is very good for small networks, but when you have the whole protein-protein interaction network, you want to look with 10,000 nodes and maybe 100,000 edges, it's very difficult to see anything. It becomes a hairball again, right? And also, if you want to overlay a lot of different types of data, like in multi-omics study, we want to overlay proteomics, gene expression, copy number, all this data to the network, it becomes challenging. I mean, you can only change the shape, color, but I mean, there are a lot size, but there are a lot you can do. So one thing we did was to use the hierarchical modular organization property we learned from the biological network, and then try to use that property. So if we can use this bar to represent the whole network, and then we can use smaller bars to represent each of these subnetworks, and then we can, each of these subnetworks can be further organized at smaller modules, and eventually we have each of the nodes located in under these modules. So as you can see, this is because the way we can do this is because the network is organized in a hierarchical modular way, right? That's what we learned from the network property. The beauty of that is now, instead of using 2 dimensions to visualize the network, we only use 1 dimension, and this way we can visualize the data in the 2nd dimension, either on the paper or in a browser. So I won't go into the detail, but we have the method to convert a network into this modular structure, hierarchical modular structure, and then to also a tool called NetGestalt that you can use to explore the data next to the TCGA data under this framework. And finally, I want to talk about, we talk about the network visualization, network-based data visualization, right? At the end, I want to talk about how we can use this network to help analyze our own data. So this is basically based on the observation that the nodes in the network are not randomly connected. Usually, genes or proteins that are functionally similar to each other are more likely to be connected to each other in the network. As you can see in this plot, like if we have a way to quantify the functional similarity between two proteins, and then we can see the protein pairs that are directly connected to each other, meaning they're one step from each other. They have much higher average functional similarity than the protein pairs that have shortest path lengths of 8 or above. As you can see, this relationship is very obvious. So by leveraging this observation, so we can come up with a few different ways, help us to do either predicting gene function or prioritizing genes in our studies. So first we can use, because we know that direct labels are more likely to share the same function, we can explore the direct interaction partners of the proteins in the network. And then the second approach is we can divide, use some graph algorithms to try to separate the networks into modules. We know there are modules in the network, right? And then we expect that the proteins in the same module will likely to share similar structure, share similar function. And the last method is called the diffusion-based approach. I think this is a very local method that you only focus on the direct relationship. This is a relatively more global method. You explore the module. But the diffusion-based approach basically try to explore the whole network as a whole system. I think this is probably a more powerful approach. I can show you a few examples to help you to understand this approach. Let's say if this is a small protein-protein interaction network, let's say, and then we know the red proteins, meaning protein have function A, and then the blue proteins, these are the protein associated with function B. And then we have other proteins in the network that we don't know what they do in the network. And then we try to use this network to help us to predict what are the functions of this network. Do they more likely to have the red function or have the blue function? And if we do a direct neighborhood analysis, so basically for each node, we can count how many red labels it has and how many blue labels it has. And through this, we can assign the function through a majority voting algorithm. So basically, if there are more red labels, then it's red, and if there are more blue labels, then it's blue. So this is very easy to implement, and you can quickly get the function of the proteins. But one limitation of this approach is that, because these three proteins, we basically have no idea about their functions at the beginning. But maybe for some proteins, like this guy, we're pretty sure it's more likely to be a red protein, but for others, it's less clear, right? So you can also think about doing this in an iterative way. So let's say you do you have an intermediate step here. You make a temporary assignment. For example, this is to pink, meaning it's more likely to be red, and this is light blue, meaning it's more likely to be blue than red. But after that, for the gray proteins, you do the recounting, and in this case, we can see this protein get to assign to a red function rather than a blue function. So this iterative process can better leverage more information than just using the original neighborhood analysis. And we can also use a module-based approach to do this. For example, in this case, we only have one protein with unknown function, and then we want to assign the function here. And if you do the neighborhood counting, and then you will see 1, 2, 3, 4 here, and 1, 2, 3 here, you will think maybe this protein is a blue protein. But if you do a module-based approach, and especially if you have a method that can allow the module to have overlapping members, and then you can have a module like this, 1, 2, 3 modules. And then this basically is a module dominated by red proteins, and this is a module dominated by blue proteins. Then you can probably think, okay, this protein might have both functions if we use this. And that is more likely to be true, right? A protein a lot of times may have multiple functions depending on what it interacts with in a specific condition. And the diffusion-based approach, this is particularly useful in gene prioritization. Let's say if you do a high throughput study and then you get let's say we do a GWAS study, we may come up with multiple SNPs or SNP-associated genes that are associated with a phenotype. Or if you do a differential proteomics analysis, you come up with 20 proteins or 10 proteins that are very likely to be associated with a phenotype. And then you want to do experiments, next step, right? But which protein to choose to do the knockout experiment? If you have 100 candidates, it's very difficult to know. So the network-based approach can help us to prioritize. So let's say these are the candidates we have, and then we can map them to the network. And after you map to the network, you can use a process called random walk process. So basically, we can imagine each node is a person. Let's say I'm a person, so I start from here, from the red node, and then if you just take random walk at each step to the next node. For example, at the first step, I can go either here or here or here or here, right? But after you go here, I have the next step, I can go here or here or here or here or here. So, but you can use some iterative updating process, and then you, at the steady state, they should end up, they have the probability to end up somewhere. So let's say if you start from here, then at the steady state, I may have higher probability to end here or here or here, right? And then we can calculate if we start from all the each of these 10 nodes, what's the probability of ending on a particular protein, and then you sum that up, and then you get the steady state probability for ending at that protein. And then I use the color shade to indicate the probability. And as we can see here, and if we start with these 10 possible positions, where we're likely to end in this area, but it's also possible to end in these areas. So anyway, I mean, for the 10 proteins, you can probably see, okay, these 4 proteins are more important than the other proteins. And also, this might sometimes help you to identify new genes not included, especially, let's say, in proteomics. We have a lot of missing identifications, right? For example, this might be a low abundant protein, and now you see maybe this is somewhat possibly important protein. So let's see some real world application of this method. So in this study, through a GWAS study, let's say, you identified a lot of genes or the SNPs associated with these genes that are potentially to be important or candidate genes for disease. But let's say you also have prior knowledge on which genes you already know to be associated with disease. Now, you have a protein-protein interaction network, and then you can map all the known genes and the new genes in the network. And then if we go through the diffusion process, we can estimate, if we just start from this node, the known disease-related genes, and then what's the probability of ending on these proteins. And then we get a new score for these proteins, and based on this process, we can rank, and you would expect the gene 3 will, very likely to be a disease-related protein than this gene 1, right? So this is very easy to understand. And also, we used this in a study for gene signature prioritization. We worked with another group, so basically to try to develop gene expression signatures for colon cancer. But we are just one group to do this study, and there are many groups, because this is an important question, colon cancer gene expression signature, and there are 7 published studies on this topic. But if we look at the gene signatures reported by these 7 studies, they don't overlap. Actually, you see very little overlap of the gene signatures reported by these studies. So then we were thinking, what is causing this discrepancy? Is it because just all these are incorrect identifications, or there are some possible other explanations? So one way to think about this is, maybe if this is a network, and each study and this is a network that is actually driving colon cancer poor prognosis, and each study may identify some of the important components in the network, but maybe they didn't get all the important nodes. But also they observe some other proteins that are not critical to this network, but they just covary with those nodes. So for example, gene signature 1 may only identify this, and gene signature 2 only identify this. So if you only do the overlapping, they have no overlap. But if you map all those signatures to the network, we will be able to identify maybe it's this region that is important. And the similar idea is, not only, this is only for mRNA-based gene signature study, right? But protein activity can be altered at multiple levels. At the DNA level, for example, this protein, the activity of the protein can be altered by mutations or copy number alterations or mRNA, the expression change or protein expression change or PTM modifications. All these can potentially alter the protein activity. And if protein activity in this important network is altered in a sample, then you are going to see potentially a phenotype, right? So that means if we have multi-omic studies, we can also map all the observations to the networks that can also help us to prioritize the findings from those studies. One example is we again collected all the gene expression signatures from the 7 studies, and also we collected all the mutations in colon cancer, and then we mapped those to a protein-protein interaction network. And then through the network algorithm, so basically it's a network diffusion algorithm we talk about. And then we get a new list of proteins that are important, not only because of their differential expression mutation, but also because of their kind of centralized location in certain part of the important part of the network. And then we were able to come up with prediction models based on those signatures. And then we were able to show that the signature you get this way has better reproducibility when you apply to a new cohort than the gene expression signatures you started from individual studies.
Because most of the network analysis, what you're doing, you're getting the information for the networks is being obtained from experimental computation and all these tools. So most of many times the interactions are transient interactions. The interactions between proteins are transient interactions, they are not permanent interactions.
Yeah, that's right.
Yeah.
That's right.
So how robust are the computational tools to pick them up? Because there are chances you may miss them out in the experimental. And how well represented are these databases as far as these interactions are concerned?
Yeah, that's a very good question. So if you go to BioGrid, for example, you download all the protein-protein interaction in the database, and that is basically the collection of all the possible interactions that has ever been reported in any of the publications. It could be in a disease state, it could be in after EGFR treatment, or it's not condition dependent. So basically you get is a map. It's just like a Google map with everything on the map, but you don't have context dependent information. So there are a few different ways to address this. For example, you can try to build your own conditional specific networks through experiments. You can do pull down or you have to do pull down in the specific condition. Or you can also try to leverage some gene co-expression information. For example, if you are interested in colon cancer, you can take a look at the colon cancer co-expression and try to overlay or integrate the co-expression information in that condition with a big protein-protein interaction network and try to get some conditional specific network. That is actually a very active research area, and the people are trying to develop algorithms to come up with context-specific interaction networks rather than just this global. But I think that still provides you a reference map that you can start to derive condition-specific networks. But that's a very important question. Yeah.
But can we predict the interactions on the basis of a primary sequence?
Well, I think, of course, you can do the prediction based on the sequence. I mean, that's, for example, one approach I talk about, the domain-based approach. And basically, if you see and I think people are start to use deep learning. Actually, some people in my lab are doing this type of analysis. They try to leverage the deep learning, and then when you have enough training models, and then you can use that as training examples, and then to see how can we use deep learning or typical machine learning approach to capture the sequence features that can help you to predict. Yes, and the sequences are also important in predicting.
So
yeah.
both memory and the 3D structure.
Exactly. Yes. Yeah. Of course. Yeah. I think people are, and there are some recent deep learning-based methods. They try to incorporate the linear sequence and also the protein structure information and some domain information as features to predict.
In today's lecture, we learnt about the importance of network visualization and interpretation of complex data. We also learned that proteins connected next to each other usually have a higher functional similarity. This forms the basis for network analysis tools. If the aim is to predict gene function, the network based methods like module based approach and neighborhood majority voting can be used to get important leads. The diffusion based approach is used to get information on gene priority in a network, and network visualization tools like Cytoscape and NetJetStart are widely used in clinical studies. I hope these two lectures and various tools which Dr. Bing Zhang showed have been helpful to you to now use your own data set or publicly available data set and try to create the various networks and visualize them using these tools. Thank you.